Hello again, Year 9. So welcome to this week's lesson on transport of molecules across cells. This is still in line with the cells topic that you have been studying over the past few weeks. So before we start, I want to say a massive well done to those of you that have been completing this work to the best of your ability each week. We have some fantastic examples of work that you've completed, and it's clear to see how hard you have been working. So well done. Remember, once you've completed the lessons, you need to send your notes as a Word document or just a photo taken on your phone to your usual science teacher. Each week there's also a compulsory Educate quiz to complete, then an optional extension or research task. So this lesson, for this lesson that we're doing today, you need a copy of the booklet Diffusion and Osmosis, which can be found on the Science Continuity of Learning webpage. You either need this printed or just open electronically. I want you to work on a piece of paper with the booklet up on the screen, or you could work on the actual Word document itself. Remember, one of these needs sending to your teacher. You'll also need a pen if you are writing your answers on paper, and it would also be good if you have a different colour pen so that you can mark and improve your answers as we go through the lesson. So just quick reminders, like I say each week: make sure your phone is turned off or put into a different room. You don't have any other tabs open on your computer. I want you to try every single question that I ask you to during this lesson, even if you're struggling. And whilst you're trying these questions, you're going to need to pause the video, attempt these questions independently, and then we'll go through the answers when you replay the video. You can always rewind and listen again if you are not sure, if you're struggling on any part of it. And remember, you can also use BBC Bite Size, so if you just type in Diffusion and Osmosis BBC Bite Size on Google, you'll be able to find the relevant web page in case you want to go look on that as well. And then finally, don't give up. Every single person doing this lesson now is capable of completing this lesson, just as long as you are working hard throughout it. Okay then guys, let's get on with it. So if you have a look at the front of your booklet, you should be able to see some do now questions. So you should either have the higher booklet if you are in 9A1 or 9B1, and then every other class should have the foundation booklet. I want you to have a go at these questions. These are all recap questions from the past few lessons. You can type your answers into the booklet if you are working electronically, or write them if you have a paper copy. The answers we're going to go through on the next slide, but for now you should pause the video, have a go yourself. I'll probably give yourself about 4 minutes for this task. Off you go guys. Right then, how did you do? So question number 1. What is the ball of cells that is formed just after fertilisation called? We mentioned this about 4, 5, 6 times last lesson. The ball of cells is called the embryo. Then what are the cells called in the embryo for question 2? They are called stem cells. Remember, these are unspecialised cells. Question number 3. As we go through these, remember you are either ticking your answers in a different colour pen, or you are adding any detail that you missed out. Question number 3 then, what happens to the stem cells as the embryo develops into a baby? They develop and they differentiate into specialised cells. So they get specific features or specific organelles which allow them to be better at their job. Question number 4, why do we need specialised cells? Because all specialised cells do different jobs in different organs in order to keep our whole organism going. And then number 5, what is meant by partially permeable membrane? So we didn't mention this last lesson, but you might have mentioned this already in year 9. I know that I've definitely mentioned it with my class. So partially permeable, if something's permeable, it means that it lets things through. Partially permeable it only lets certain things through. So it's a membrane that allows some substances through but not others, depending on how big they are. So what a membrane has is tiny little holes, and they are small enough to let some things through but not big enough to let other molecules through. What are you going to be learning today then? So during this lesson we are going to be describing how substances move in and out of cells by the process of diffusion, and we'll be giving some specific examples of that. We're also going to have a go at describing and explaining factors that can affect the rate of diffusion, so how fast diffusion happens. Then we're going to move on to describing what osmosis is and giving examples of where osmosis happens in living organisms. And then also we'll be popping a little bit of key science skills in there. So we'll be having a go at identifying variables to change, measure and control to test a hypothesis. And remember, a hypothesis is just a scientific prediction. So first things first, let's have a look at what diffusion is. So you met diffusion before in the Year 7 cells and particles topic. You also experience it in everyday life. When someone's spraying perfume or you smell what is cooking for dinner downstairs. So diffusion is just the movement of molecules, liquids or gases from an area of high concentration to an area of low concentration. High concentration just means where lots of particles are clumped together in a really small area or small volume, and they move to a lower concentration where there are less particles and the particles are much more spread out. Why do the particles move then? Why don't they just stay where they are? So diffusion happens because there is a concentration gradient. And I always think of this like if you was on a bike at the top of a really steep hill, so you're high concentration, and then what happened is you lifted your feet off the floor, didn't pedal. What would happen is you would move down to the lower concentration. A concentration gradient just means that there's a difference in concentration in two different places. So in one side of the room there's much more particles compared to the other side of the room. When the two sides are equal, the particles don't stop moving, it's just that their movement in either direction is approximately equal. So concentrations remain constant, or more or less constant. There are lots of examples of diffusion happening in living organisms, and I'm just going to go through a few examples on this slide here now. Remember, when diffusion happens in the human body, it's not particles moving from one side of the room to another side of the room. It's particles moving from either outside of the cell into a cell, or from inside of a cell moving back out of the cell. So some examples then of substances that move by diffusion in the human body is oxygen. So oxygen moves from the lungs into the blood. So when we breathe in, we breathe the oxygen into our lungs, goes right down to our alveoli, and then surrounding our alveoli is bloodstream, and what happens is oxygen moves out of the alveoli into the bloodstream. We also have carbon dioxide, which moves in the opposite direction, so from the blood cells into the alveoli cells. We have glucose as well, which moves from the blood into our cells, so it's moving from one cell from a blood cell into another cell. And then also digested food particles from the small intestine. So when you eat something and you digest it and it gets into the small intestine, around the small intestine is the bloodstream, so the blood cells again, and what happens is the digested food particles will move from the small intestine cells into the bloodstream. So this is an experiment that we would have done if we were in school. So we take some starch and sugar solution. Sugar is the same as glucose, so starch is a big molecule made up of many sugars combined together, and then glucose is much smaller. So we take some starch and sugar solution and we put it inside a tube. Around this tube is a partially permeable membrane. Remember, that means that it lets some substances through because it's got tiny holes which small substances can fit through, but not other substances, so it doesn't let other big substances through. So what happens then is we leave the experiment for around 15 to 20 minutes, and then the distilled water around the tube is tested for starch and sugar, so that distilled water inside the container. I want you to think about, what do you think would happen and why? Where do we think we'd find the starch? Where do we think we'd find the sugar? Would we find it still inside the tube, or would we find it in the distilled water? So pause video now, give yourself a couple of minutes to have a think about the particle movement, what concentration gradients there are, and think about particle size. Okay then, so after around 15 to 20 minutes there would be glucose in the distilled water, so glucose would there'd still be some glucose inside the tubing, but it would also be inside the distilled water as well. Some of it will have moved out, and this is because we had a concentration gradient of glucose. We had a high concentration inside the tube, so it moved outwards into the distilled water until it was equal on both sides. Remember, the glucose could fit through because it was much smaller than the starch, so it could fit through that partially permeable membrane through the tiny holes. The starch, however, still has a concentration gradient because there's a high concentration of starch inside the tubing, but the starch cannot fit through the partially permeable membrane. The holes are too small for the starch to fit through. So the starch will just stay inside the tubing. Time for a learning check then. So if you look underneath your Do Now task in your booklets, you should be able to see questions 1 to 3. I'm going to have a go at the questions, and there's several things on this slide to help you. So there's words for you to try and include in your answers. I want to complete these answers in full sentences, and to help you do that, I've included some optional sentence starters that you can use if you feel like you need to. Question 1 and 2 is to just give examples of diffusion. Question number 3 asks you to explain some things about that experiment that we just went through. So the first one asks you to describe what can be found in the distilled water. So you have a sentence starter for this, but you can also flick back to the previous slide where we went through this. And then question 3b is probably the most difficult, but this one is asking you to relate that experiment to the digestive system. So that experiment was purposely set up in order to mimic the human digestive system. So I want you to have a look at the 3 things that it includes in question 3b. So the tube, the distilled water, and then the starch and sugar solution. And to have a think about what they could represent in the human digestive system. And the first one I've done for you to help you. So 3b, the tube was mimicking the small intestine. So having a think then about what the distilled water is replicating and what the starch and sugar solution is representing. We should have attempted these questions by ourselves first, and now you should have a different colour pen in order to correct or add in any detail to your answer. So question number 1 asks you to give examples of diffusion in real life. So we talked about this right at the start of the lesson. So smelling someone's perfume, smelling something cooking from another room, smelling the sewage works as you drive past it in the car are all examples of diffusion in everyday life. Question number 2, examples of diffusion in the body. Remember we talked about 4 key things that move by diffusion. We have oxygen, diffusion from the lungs into the blood. We have glucose, diffusing from the blood into the cells. Carbon dioxide, which moves from the blood by diffusion into the lungs. And we also talked about digested food molecules as well. Question number 3a was where we had to describe what and explain what had happened in that experiment. So after 15 to 20 minutes you would find that glucose would be present in the distilled water, but starch would not be present in the distilled water. Remember, starch is a much bigger molecule. So there's a higher concentration of glucose inside the tubing, so it's going to move by diffusion from the high concentration inside to the lower concentration outside the tubing in the distilled water. And glucose is small enough to fit through the pores in the membrane, so it moves out by diffusion. But there's no starch in the surrounding water, because even though there's a higher concentration inside the tube than outside, the membrane was only partially permeable, so the pores, the holes in the membrane, were not big enough to let starch through. So correcting and adding detail in a different colour pen. And then finally, question 3 was the starch and the sugar. So the starch and the sugar represented the food that had been eaten and dissolved in the small intestine. So green pen in your answers now, guys. This then, once you've done that, I want you to try 2 tea bags, one in cold water and one in hot water. Is there a difference in how quickly the colour moves out of the bag into the water? And why do you think there might be a difference? What's the explanation for this difference? Have a think about that. So that home experiment sets us up really well for this next part of the lesson, the rate of diffusion, how fast diffusion happens. So the speed, the rate of diffusion, can be affected by 3 main factors. The first one being the concentration gradient. So if there's a higher concentration inside the tube at the start, then the glucose would have moved much faster. So if there were 500 glucose molecules in the tube at the start compared to another tube where there was only 100 glucose molecules, it would be much faster in the first tube because there was a much higher concentration gradient. The second thing that affects the rate of diffusion is the temperature. So particles have more energy at higher temperatures, so they can move faster which means it speeds up the rate of diffusion. And then finally, the surface area. So if you have a bigger surface area, you have more space for the particles to move in or out of a cell or a membrane, across a membrane, so diffusion will happen faster. Osmosis simply means the diffusion of water. It's movement of water from a high concentration to a low concentration through a partially permeable membrane. It can be a bit confusing talking about a high concentration of water. This just means a really dilute solution. Like if you were making a glass of Vimto and you only put a bit of Vimto in but lots of water, that would be a high concentration of water and very dilute. Of course, the most dilute solution that you can get of water is distilled water. So pure water is the most dilute, the highest concentration of water you can get. So here we can see osmosis in action. Solute just means something that has been dissolved in water. So again, we can see a concentration gradient across the membranes that line in the middle. There's a higher concentration of water on the left. The solute cannot move, so the water particles move. Solute is too big to get through the partially permeable membrane. It is important to realise that water would move in both directions because particle movement is random, but more water would move right than it would move left. So there would be an overall movement from left to right because there's a higher concentration of water on the left at the start. So this is an experiment similar to the one that we looked at earlier. I want you to think about what you would expect to happen here. We've got starch in the middle of the tubing and then we've got water outside. Look at the concentration of water. Where is there a higher concentration of water? Is it inside of the tubing or outside of the tubing? We already know that starch cannot move out as it is too large to fit through the partially permeable membrane. So what's going to move? Pause the video and have a think. So, did you get it right? It is the water that would move, and it would move from the beaker because we've got distilled water, so it's very, very high concentration of water in the beaker. So water would move from the beaker into the bag because water is small enough to fit through the partially permeable membrane, and this would happen by osmosis. The level of liquid would rise inside the bag. Osmosis can affect cells in our body, and this is why it is so important that we stay hydrated. This is what happens when people become dehydrated. So in the top diagram, the cells, probably not taken from humans, probably taken from an animal, have been surrounded by salt solution. And in the bottom diagram, the cells have been surrounded by distilled water. What has happened to the cells and why? Thinking about the shape of the cells, thinking about the content of the cells, what has happened? Which one would be an example of when dehydration happens? Stop the video and see if you can explain what has happened using the idea of osmosis. You don't need to write this one down, just have a think about it to yourself. So, did you get the right idea? The first one, when salt solution was added, there was a higher concentration of water inside the cells compared to outside the cells. So what happens is water moved out by osmosis. This meant that the cells shrank and start to shrivel up. And this is what happens if you become dehydrated, extremely dehydrated. This can cause cell death. The bottom image shows that the cells have swelled, and this is because there was a higher concentration of water outside the cell compared to inside. So water would move into the cell and it could cause the cell to burst because it becomes so full with water that it bursts. And this can also be dangerous. In both of these images, water will have moved by osmosis because that is the only way that water can move. Back to your booklets then. So, you've been given 3 images, A, B and C. A shows normal blood cells. I want you to have a think about what has happened in B and C using the idea of osmosis. Try and write your explanation of what happened in full sentences in your booklets. Pause the video while you have a go at this and we'll talk through them in a second. Well done for having a go at that, guys. So, in B, we can see the blood cells have swelled up. This must mean that water has moved into the cells. There must have been a higher concentration of water outside the cells than inside, so they must have been placed in water or a very, very dilute solution. The opposite has happened in C. The cells have shrunk, so water has moved out of the cells as there is a higher concentration of water inside the cells compared to outside the cells at the beginning. They must have been placed in a concentrated solution, for example, salt solution. In both B and C, we can tell that the cells are extremely unhealthy. So both of these, too much water and too less water, can be extremely dangerous. The final part relating to this week's content then is a learning check. I want you to have a go at answering these questions. Don't worry about them being in full sentences. These are just quick recall questions to see what you've learnt this lesson. Think through the answers. You don't need to write them down, but you might find it helpful to write them down. And then, if you give yourself about 3 or 4 minutes to answer these, we'll go through the answers on the next slide. This is just bringing together your learning for this lesson. So hopefully you didn't find those questions too difficult. Remember, if you have struggled with it, please go to BBC Bitesize on the cell transport, diffusion and osmosis page. So question number 1, by what method does oxygen move from the blood into cells? It was by diffusion. Number 2, what does partially permeable mean? Let some molecules through, but not all molecules. 3, what means by osmosis? It is water. Number 4, what can happen to cells when osmosis happens? They can either swell or shrink. Number 5 was factors affecting how quickly diffusion happens. Temperature, concentration gradient and surface area are all things that affect how quickly diffusion happens. Question 6, why do diffusion and osmosis happen? This is because of concentration gradients, concentration differences. And then number 7, what does a high concentration of water tell you? It tells you that the solution is very, very dilute because it's got lots of water. So check you got the answers correct. Some of these are going to appear in the Do Now part of next lesson. So, I also told you, as part of this lesson, we were going to be practicing our key scientific skills, identifying variables to test a hypothesis, to test a scientific prediction. So the fluency test is on page 4 of your booklets. I mean, to have a go at that fluency test, give yourself about 10 minutes to get through as much as you can of that, and then you can mark it using the mark scheme that has also been uploaded onto the continuity of learning science page. After you've had a go at the fluency test, there's also some exam questions that you can try for yourself as well. If you think it's too much to do right now, maybe do half an hour of science tomorrow where you just do the exam questions. It's all about making sure you're having enough breaks, but then also spending enough time doing your work as well. If you want a tip, what I've been using to help me focus and do work at home is the Pomodoro technique. So if you just Google Pomodoro technique, you can find a timer on YouTube, and what this does is it times you for 25 minutes, so you do 25 minutes solid work, and then every 25 minutes you get 5 minutes to just go and have a break, do what you want, go on your phone for 5 minutes, do a little lap of the house to give yourself a break away from that work. And that's been working really well for me, so I'd recommend it. Once you've finished the exam questions and the fluency test, there's also that extension, that research task, which we've attached if you'd like to do it. And then remember, you've got your Educate quiz to do as well. This is due at 8 AM on Monday. So with that in mind, I would like to say a massive well done. That's the end of this week's content. Well done for all of your hard work so far. You are doing absolutely fantastic, Year 9. Please keep it up. Remember, if you've got any issues or any questions at all, just get in touch with your science teacher. Enjoy the rest of your week, guys.